In our next section, we'll discuss the physiology of axonal transport. In this section, we'll discuss axonal transport. As described earlier, the length of the axon relative to the cell body poses some specific problems that the cell has to solve, particularly in terms of getting important cellular components from the cell body out to the synaptic terminal. For example, most neurotransmitters have their synthetic machinery packaged into the synaptic swelling. Again, as an example, catecholamines have their synthetic machinery, tyrosine hydroxylase, etc., all localized in the synaptic terminal. However, none of these enzymes are synthesized there. They're all synthesized in the cell body and have to be transported down the axon to their site of action. The way that the cell handles this type of long-distance transport is via a microtubular system, sometimes called the superhighway of axons, that allow these specialized cells to have communication between the cell body and the synaptic terminal. Shown here is how this transport actually occurs. Retrograde transport, for example, is important for the transport of spent synaptic vesicles from the synaptic terminal back to the cell body. Proteins such as dynein and kinesin are important for this long-distance transport to occur. In the specific case of retrograde transport, dynein, which is a molecular motor, that is, it is a protein that can move along this microtubule, will carry the spent synaptic vesicle along the microtubule back to the cell body. There is directionality to these microtubules, such as there is a positive and a negative end, and the dynein will carry the spent synaptic vesicles from the positive end to the negative end back to the cell body. Kinesin, by contrast, will carry other synaptic cargo, things as described earlier, like proteins that are important for the synthesis of neurotransmitters, mitochondria, etc. This will travel along the microtubules with a protein known as kinesin, another form of molecular motor, from the negative to the positive end. Now, anaerograde transport comes in two different types. There is fast anaerograde transport, as shown here, and then there is slow anaerograde transport. Now, fast anaerograde transport and retrograde transport, of which there's only one type, both operate under relatively fast speeds, approximately 200 millimeters per day. Slow anaerograde transport operates at a much slower time scale, approximately 1 millimeter per day, and the mechanism by which this occurs is not well understood. What's important for you to recognize is the difference in the type of cargo carried in fast anaerograde versus slow anaerograde transport. Fast anaerograde transport is important for the daily housekeeping jobs that a synaptic terminal has to complete, such as the synthesis of neurotransmitter, the synthesis of ATP, etc. But when an axon is damaged and has to be reformed, or during the initial formation of an axon, certain other molecules have to be transported down the axon. Things like molecules that are part of the cytoskeleton, things for the growth of the synaptic terminal, have to be carried down to the axon, down to the terminal, and this transport is called slow axonal transport. And again, this is an unknown mechanism that travels at approximately 1 to 2 millimeters per day. Now, it's important to recognize that several of the neurotropic viruses utilize the fast axonal transport mechanisms in order to cause their pathology. An example here would be varicella. Varicella, as we probably all already know, is a disseminated disease early in life, but then takes root in the dorsal root ganglia and may live there for decades before re-emerging. When it re-emerges, it will utilize one of the fast axonal transport mechanisms in order to invade the skin, and as shown here on the figure on the right, is a vesicular eruption in a single dermatome. The eruption may last a period of 1 to 4 weeks, and then ultimately the eruption will cease and the organism will again utilize fast axonal transport to take root again in the dorsal root ganglion. This concludes our discussion of the physiology of axonal transport.